Welcome to today's explainer. We are jumping right into what I can only describe as a massive biological plot twist. Seriously, we're looking at a fascinating paper that recently dropped in Nature Microbiology, and it completely flips our understanding of disease-causing proteins right on its head. So let's just dive right into this wild premise. Could the exact same proteins responsible for deadly brain diseases actually hold the cure to antibiotic resistance? I know, it sounds completely paradoxical, right? Especially with the global superbug crisis we're facing right now. But honestly, that's exactly the mystery this research set out to solve. Now, traditionally, prions are the architects of some terrifying, absolutely incurable conditions like mad cow disease. They're notorious for protein misfolding and neurodegeneration. If you look at the standard story of biology, well, prions are the ultimate villain. But here is where the profound irony of the researchers' hypothesis comes in. They basically asked, what if this villain is secretly hiding a hero? They had a hunch that these notoriously sticky, aggregation-prone proteins might actually encode cryptic, life-saving fragments hidden deep down inside their amino acid sequences. Section 1. Mining with Deep Learning, the Apex 1.1 Sieve. To actually find these hidden fragments, scientists obviously couldn't just peer through a microscope. They deployed an artificial intelligence framework called Apex 1.1 to act as this giant molecular detective. And just to wrap your head around the sheer scale of this search, the AI started by analyzing over 19 million unique candidate peptide fragments. 19 million! And these were meticulously extracted from nearly 3,000 curated prion and prion-related proteins. But through the predictive filtering power of deep learning, the APEX framework drastically whittled that massive haystack down to just 1,179 prime candidates. The neural network mathematically predicted that these specific select sequences would pack a serious antibacterial punch against a really tough panel of 11 different pathogens. So the researchers actually coined a brand new term for these 1,179 hidden candidates. They called them Prionins, and this concept kind of anchors our whole scientific mystery today. Prionins are basically encrypted antimicrobial peptides discovered within prion proteins, and they come from over 1,000 different organisms. We're talking fungi, metazoans, even unicellular eukaryotes. Section 2. Meet the prions, deciphering the encrypted code. Okay, so let's see how these computer-generated predictions actually stack up when compared to the real world. When they mapped it all out, the sequence space projections showed that prionins occupy a totally distinct physical-chemical landscape. Basically, compared to the antimicrobial peptides we already know about, prionins are generally longer, they're more positively charged or cationic, and they're way more water-repelling or hydrophobic. So this proves the AI didn't just spit out copies of what we already knew. It found completely unexplored sequences. Of course, computer predictions are really just the starting line, right? To see if the math translated to reality, the researchers went into the lab and physically synthesized 75 of these highly diverse, top-ranked peptides. And wow, the results were absolutely striking. Out of the 75 synthesized peptides, 59 successfully inhibited at least one clinically relevant bacterial strain. And yes, that includes multi-drug-resistant isolates. On top of that, 42 of them showed highly potent minimum inhibitory concentrations, with the activity heavily concentrated against gram-negative bacteria. I mean, that is an incredibly high experimental hit rate to get directly from AI predictions. So how do they actually work? Well, they found that these prions attack like a tactical strike team. Step 1, they transform from unstructured, loose shapes in water into highly ordered conformations the second they engage their target. Step 2, they permeabilize the bacterial outer membrane. And step 3, they just completely collapse the cytoplasmic membrane potential. Essentially, they are lethal membrane disruptors. Now, obviously, an antibiotic is completely useless if it kills the patient, along with the bacteria, so they had to run a toxicity check. And thrillingly, the researchers found 16 active prions that showed absolutely zero toxicity to human embryonic kidney cells or human red blood cells. They selectively destroy the bacteria, which proves that this prion-derived activity isn't inherently toxic to our own mammalian cells. Section 3. Putting prions to the test, the mouse model. All right, the highest stakes part of the explainer. Let's see if these encrypted codes can actually cure real infections in living subjects. The team selected 2 lead contenders based on how potent and safe they were. We have Prionins 7, which is derived from a fungus, and Prionin 38, derived from a microscopic worm called C. elegans. They pitted both of these against a really tough Acinetobacter baumannii skin infection in a mouse model. So here is how they structured the trial. The mice were infected, and then just 1 hour later, they received a single topical dose of the prionin at 10 times the minimum inhibitory concentration. And this is key. The mice showed absolutely no treatment-associated weight loss, meaning 0 negative side effects were observed from the therapy. And the breakthrough here is pretty undeniable. By day 4, Prion 38 maintained a massive 3-log reduction in the bacteria. To put that into perspective, that is essentially wiping out 99.9% of the bacterial burden compared to the untreated group. What's even wilder is that its performance tracks almost identically with polymyxin B, which is a standard, highly potent clinical antibiotic. Just let that sink in for a second. An encrypted peptide originally from a worm's prion protein just cured a superbug infection. Section 4. A new frontier in biology. Redefining the villain. Ultimately, what you should really take away from this explainer is that deep learning just proved something incredible. These very proteins we've always associated with neurodegeneration are an unexpectedly rich reservoir for future antibiotics. The author stated it perfectly, noting that this expands a growing view that antimicrobial activity can literally be hidden within proteins not canonically annotated as immune effectors. It suggests that these cryptic, aggregation-prone sequences can actually birth defensive, life-saving fragments. Honestly, it completely shatters our standard view of biology. We always thought prions were just disease vectors, the bad guys. But if deadly prions are secretly housing encrypted antibiotics, it leaves us with one massive, lingering question to ponder long after this explainer ends. What else might our biology be hiding in plain sight?